All right, well, hello everyone, and welcome back to the show. It is I, your favorite Jackson, the good Jackson. And joining me today, we have Nesslig, one of my editors. Welcome back.
Hello. Good to be back.
Good to have you. You're going to be telling us all about photosynthesis reaction centers today. Yay!
That's right. But I am not Canadian. That's the only error I have to correct.
And we also have Peter, who is my host and co-host. Welcome back, Peter.
It's nice to be back. It's been a while, Jackson.
Yes, it has.
So, what were you doing last week?
Last week, I was driving alligators down to the Louisiana coast, and I know that sounds like a bit, but it's not. I really had to drive down to, I believe it's pronounced Grand Chenier. I think that's pronounced. And we had to deposit, one of my coworkers and I had to deposit some baby gators, and then we had to pick up some more gators from the place where we deposited them and bring them back to the aquarium.
Mm-hmm.
So that was fun.
And
how do you call baby gators?
How big are they?
How do you call them? Like
you can call them anything.
I know marsupial babies are referred as joeys, right? Like marsupial babies are referred as joeys. I'm not sure there's a special name.
I don't know. Gator bites.
Babies.
I don't know. Whatever you want to call them, I guess.
You can call them anything. They won't come if you call them anyway. So
that's true. They're famously bad at following instructions.
Jackson, you know that I work with a Hollywood actress every week. And so I pitched the idea of what you were doing, and apparently there might be a studio picking it up because I saw this.
Oh, that's really funny. I have a cat who's trying to climb up me right now.
Crocodile young wheat.
Yes, because the previous one was Dundee, so you have to have 2 syllables.
Yeah. Yep. That was me.
Anyway.
Although there's more alligator young wheat.
Yeah.
Oh,
a slight mistake. It's an alligator, not a crocodile. And to know more about that, we did a series on what the difference is between crocodiles and alligators a while ago.
See, but you have crocodiles in Australia, where Dundee's from. Mm-hmm. Here in Florida, we have gators.
That's not a knife. This is a knife.
Yeah. Okay. Now you're talking about knives. I'm gonna leave, and I'll leave it to you two. Now it's getting too dangerous if you're gonna pull out knives.
That was funny, Peter. Thank you.
Hi.
Yeah. So we had to take the gators, because I was thinking about it after I messaged you guys. I was like, they're not going to believe me when I tell them what I have to do. So the most Louisiana thing I think I've ever said. So
to be honest, the first time I saw it, I was thinking, well, you're referring to people from Florida, just like you're calling them alligators. I thought you were referring to that.
No, some little baby gators, but all right. Well, Nesslig, it's your show, and I will turn it over to you.
All right. I'll also, as always, feel free to interject, or if there is any comment, any questions in the live chat, and I will, of course, pause for that. Okay, you bring up the slides, Peter? There you go. Also, can you make it? Yeah, thank you. You can make it bigger for us as well. So this is the 4th chapter in our series, and we are crawling to the series because I am also busy with other stuff in my personal life. So I'm trying to make some slides to complete the series, but now we are at the first chapter or at the fourth chapter, but in the first time we are going to discuss the evolution of photosynthesis. And there are several aspects of photosynthesis that we have discussed before, like we have discussed the chlorophyll, the proteins that make up the electron transport chain of photosynthesis, and also, of course, the Calvin cycle, the metabolic cycle that draws out or draws in CO2 and converts that into biomass. But today we are going to focus on the evolution of the reaction centers of photosynthesis. Our next slide. So yeah, the first chapter was on the sun eaters, where we discussed the different types of organisms that perform photosynthesis. And in chapter 2, we discussed how the oxygen left behind by photosynthesis changed the whole world. And also, and in the previous chapter, chapter 3, we discussed the process of photosynthesis itself in great detail. And again, today, it's all about the evolution of reaction centers. My plan is to do the evolution of carbon fixation, or specifically the Calvin cycle in the next chapter. And then the last chapter will be on the evolution of pigments, like chlorophyll. But I'm not sure when I am getting around that, because I'm very busy right now. So
That's
all right,
totally
next slide.
fair. I understand.
Next slide. Yeah, so in this chapter, I have also some series on some sub chapters planned out. So the first, we are going to do a quick recap on photosynthesis, basically a rundown of what we discussed in the previous chapter, just to as a quick reminder of what photosynthesis is all about. If you want to know more about the details of that, just go back and watch the previous chapter on that. I will also discuss some things about the details of eukaryote, or the evolution of photosynthesis in eukaryotes, and then we move on to the evolution of photosynthesis in bacteria. Then we move on to what life does in the dark without photosynthesis, and that forms the basis for us to discuss the specific details of the evolution of reaction centers, the specific topic of this chapter. And at the final notes, we are also going to discuss the origin of the oxygen evolving complex, or the OEC. Next slide. So yeah, first part, the recap of photosynthesis. Next slide. So again, the photosynthesis starts with getting electrons from water molecules. The water molecules are the electron donors, and water is it's difficult to get electrons from water, because like you may have known about electrolysis, it takes electricity to split water apart back into oxygen and hydrogen gas. But here, photosynthesis does it by using the energy of sunlight. So what happens is, within the first protein system, called photosystem 2, you have a group of 4 chlorophyll A molecules, which we call P680. And P680 refers to the fact that the wavelength of light that is most absorbed by this 4 chlorophyll complex, the wavelength is 680 nanometers. Hence why it's called P680. And when that is excited by a photon of light, it donates an electron down the electron transport chain, and then left behind the P680 radical, after it donates an electron, it becomes a radical, a positively charged radical. And this is strong. This is an oxidizer that is strong enough to pull electrons from H2O. There's a problem with this, because the full oxidation of H2O to oxygen takes 4 electrons per molecule. You need to have, and P680 is only able to accept 1 electron at a time. And this mismatch between 1 electron transfer and 4 electron transfer is solved by the oxygen evolving complex, or the OEC. And you can see a description of exactly what it does in the images right there. You have on the left hand side, you see the oxygen evolving complex with the water molecules and oxygen and 4 electrons. And then in the right hand side, there's the Koch cycle, which describes the whole process of how the oxygen evolving complex works. But I will bring up more details about that later on. And then after getting these electrons from water, and then the PS2 system, or photosystem 2, the photosystem 2 donates the electrons onto another molecule called plastoquinone, or PQ for short, and that turns it into plastoquinol after it accepts 2 electrons. And plastoquinol is PQH2. It's basically an electron carrier that moves through the lipid membrane of the thylakoids within the chloroplast. Next slide. All right. And then the plastoquinol, or the PQH2, it moves through the lipid membrane onto the next protein complex called cytochrome B6F. The PQH2 donates 2 electrons, but each electron goes down 2 separate paths. One goes through the Q cycle. I discussed it in detail in the previous chapter. It's just a way to pump more protons from the stroma side to the lumen side, and that generates a proton gradient, which will become important later on. And then the other electron is donated onto another protein called plastocyanin, and this plastocyanin protein is soluble. It moves within the lumen side of the cell. Next slide. And then plastocyanin binds onto the next photosystem, photosystem 1. And the reason why photosystem 1 is called 1 is because it was discovered first. Photosystem 2, the previous one, was discovered later, even though it is the start of the chain, but still. In photosystem 2, you have another complex of chlorophyll A molecules called P700. And that like the positively radical P700 accepts the electron from plastocyanin, becoming a neutral P700. But then when P700 absorbs light energy from sunlight, it donates the electron and it turns back into the P700 positively charged radical. And the final, and then the electron donated by P700 goes down another series of electron donors, and the final electron donor, in this case, or the terminal redox center in PS1, is an iron sulfur cluster, or 4Fe-4s. It's made of 4 iron and 4 sulfur atoms. Also important to note is that unlike the previous photosystem 2, there are 2 symmetrical paths that the electron can take within the PS2. As you can see here, maybe it's difficult to see, but I have drawn out like 2 paths within the PS1 system with the little arrows. But in the PS2, there is only 1 path the electron can take. There's a method. But in PS1, it can take 2 alternative paths, which are symmetrical anyway. All right, next slide. And then PS1 donates the electron onto another protein called ferredoxin, or FD. This ferredoxin contains another iron sulfur cluster, but in this case it's only made of 2 iron atoms and 2 sulfur atoms. And then this donates it onto another protein, the ferredoxin NADP reductase, or FNR for short. And FNR uses the electrons to reduce NADP into NADPH. And that's the final electron acceptor of this, technically not the final electron acceptor, but for now it is the final electron acceptor. It turns NADP plus into reduced NADPH. Next slide. Also, again, a reminder is that since the PS2 takes electrons from water, one of the products of the PS2 is oxygen, but another product is the protons, the hydrogen plus ions, which are left behind in the lumen side. Also, we have the cytochrome B6F complex, which translocates protons, or H+, from the stroma side onto the lumen side, and that generates a proton gradient. This makes the lumen site very acidic and the stroma site alkaline by comparison. The lumen site is low pH and the stroma site is high pH. That proton gradient powers a very famous enzyme called ATP synthase. That's powered by this proton motive force. When protons are, again, the ATP synthase allows the protons to move back from the lumen to the stroma site, and that turns the ATP synthase. And that turning of the ATP synthase catalyzes the conversion of ADP into ATP. And as you may have heard before, ATP is the energy currency of the cell. Next slide. Before I move, sorry, can you go back for one moment? One other note I can make is that there was a scientist in 1961. He proposed, Peter Mitchell proposes the chemiosmotic theory that explains the coupling of electron transport and ATP synthesis. That was a very controversial suggestion way back then, but eventually was proven right that there was this ATP synthase coupled the electron transport and the production of ATP. All right, next slide. And then the NADPH and ATP produced from photosynthesis, that powers the Calvin cycle. And the Calvin cycle is responsible for converting CO2 into biomass. And there's a whole complicated chain of chemistry going on in the Calvin cycle, but I won't go over the details of that process. I have covered the details of that process in the previous chapter. All right, next slide. Also, one thing to mention is also that the PS2 and the PS1 systems, they also have other proteins surrounding them, which are called light harvesting complexes. Next slide. So this is also something I discussed before, but just another recap. This super complex of the photosystem 2, in addition to the core of the complex, the PS2 core, you also have these light harvesting complexes. There are two types. You have the major light harvesting complexes, which are trimeric, composed of 3 polypeptides, and you have also a minor antenna complexes in purple colored right there. And this super complex with these proteins is the most common super complex found in land plants. And we will discuss the variety of these super complexes when we get to the discussion surrounding eukaryotes and also bacteria. They are very different. Some of them have very different light harvesting complexes. Next slide. And also the light harvesting complexes work by increasing the ability to capture light. So light does not have to strike the core of PS2. It can first strike a light harvesting complex right there, and that energy is transferred from one pigment to another pigment to another pigment, and eventually ending up into the photoactive redox center, or in this case, the redox center is P680 that I mentioned before. Also, in addition to chlorophyll A, you also have chlorophyll B, beta carotene, and also xanthophiles. These accessory pigments increases the bandwidth of light absorbance. Chlorophyll A only absorbs light into certain ranges, but with these additional pigments, you just capture more light, basically. Next slide. Also, the PS1 system also has a super complex, but not as big as the PS1, but still pretty impressive, of course. And it also has like 4 light harvesting complexes associated with the core PS1 system. All right, next slide. And again, the same principle applies here with the transfer of electrons. Next slide. So another thing to note about these mega complexes is that they change conformation based on different conditions. So there is a regulatory enzyme called PSBS. Under low light, it is a dimer, but it's not active. But when the light intensity increases and the lumen becomes very acidic, the PSBS enzyme becomes protonated. So protons attaches onto the PSBS enzyme, and that splits the PSBS into monomers. And the monomers are active. They now begin to regulate the light harvesting complexes, and that causes them to switch into a different state. They go from a light harvesting mode to a energy dissipation mode. Basically, this is a way to avoid overloading when light intensity is too high. And they also have a very peculiar process called the Xanthophyll cycle, where so in the light harvesting mode on the left-hand side of the image, you have a pigment called violoxanthin that absorbs light. It absorbs light and donates the light onto the redox center. But then when light intensity is high, that violoxanthin is converted into another pigment called zeaxanthin, and that does not donate light energy, it dissipates it as heat. So this is a way to, again, avoid overloading. If too much light energy is exposed, then zeaxanthin is a way of dissipating the excess energy. Next slide. And also one other process is that some of the light harvesting complexes become phosphorylated by these regulatory enzymes. Sorry. I have to go back up. So the PS2 system reduces the PQ to PQH2, as I mentioned before. Under high light intensity, the PQH2 is too much of that is produced, and this, when too much PQH2 is present, it will cause the cytochrome B6F complex to activate a kinase enzyme called STN7, and that will phosphorylate the light harvesting complexes of photosystem 2. And the reverse process is mediated by a phosphatase. But when the light harvesting complexes are phosphorylated, they detach from the PS2 system, and they then move and aggregate into different structures to dissipate more excess energy. Next slide. And at the macroscopic scale, you can see that this different states causes the organization of these proteins to become very different. So on the low light condition, these super complexes form like or mega complexes. They form like almost a crystal structure, like a very regularly repeated structure from one superstructure to another superstructure to another superstructure complex. But then when light intensity is too high and these light harvesting complexes become to disassociate, then it goes into a disorganized state that is less efficient to capture the light, of course, because under high light conditions, the plant does not need the excess energy. It wants to avoid overloading. All right, next slide. This also has an effect on the very organization of the thylakoid itself. So, as another recap, the thylakoid is consisted of like these membrane structures called grana, which are layered. And these, under low light condition, these grana are highly stacked. They make a very firm contact between each other. But when light intensity gets too high, the grana become loose. They almost detach from each other, and and also the grana contain. They also begin to contract as well to reduce the interception of light as well. Again, there are many different ways of the plant to avoid absorbing too much light when light intensity is too high. Next slide. And here's another image to illustrating the process. So on the low light condition, on the left hand side, the thylakoid looks like this. But when light intensity gets too high and the light harvesting complexes become phosphorylated, then the grana shrink in size, and they also become less firmly stacked as well. Next slide. So as you may have heard before, there is a very common argument, also a very common quote cited before by creationists that say if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. But then that is the point where the creationists stopped, because Darwin also said right after that, but I can find no such case. But of course, creationists nowadays want to cite many different systems which they believe is so-called irreducibly complex. And of course, photosynthesis has often been used as an example for this. Although by most creationists not. Ironically, they more commonly use like ATP synthase
Yeah,
or
I
flagellum. Yeah.
I literally can't think of like any
Right.
time I've heard creationists talk about photosynthesis. You remember when we
Yeah.
covered Frank Turek and he said evolutionists don't talk about
Mhm.
plants? It's like,
Right.
sir, the call is coming from inside the house.
Mhm.
Which is funny because they have a few plant people over there, like Sanford. John Sanford is
Yes.
a plant genetics guy. So yeah, no, but he never
Mhm.
talks about plants. So
Yeah. But the only creationist I even know is like Otangelo that talks about photosynthesis a lot. That's the only one that
well,
I'm aware of.
sure, but I
But
mean, he's like
he's not a
always
major player, of
spamming.
course.
Yeah, he's always like
Mhm.
spamming his blog posts, which are just like ripped from Wikipedia. So
Oh yeah, yeah. Mm-hmm. All right. Yeah. Next slide. And yeah, there's also like not only are the super complexes that I just mentioned also very organized. There's a whole series of processes that are interlocked in or seemingly interlocked, but we will discuss that later on as well. Or maybe they are not interlocked as they seem. So you have to, for example, not only do you have the electron transport chain of the proteins embedded in the membranes of the thylakoid, you also have the Calvin cycle as well. But we will discuss those later on. Next slide. And if you move even further to a larger scale, you also have complex structures in complex cellular organizations like the thylakoids within the chloroplasts. Next slide. And you also have complex ecological organizations that photosynthesis seems to be, at least not too long ago, like within a lifetime even. Photosynthesis was believed to be essential to life. Without photosynthesis, you cannot have life because photosynthesis is the basis of the ecosystem. But again, we will discuss more about that later as well. Next slide. So now you move on to the discussion on eukaryotes and how photosynthesis has evolved and changed within that domain of life. Next slide. So again, a recap from the first chapter. This is a basic overview of eukaryotes and which eukaryotes can perform photosynthesis. So we have one group called the Archaea plastids. And this group inherited the primary endosymbiosis chloroplasts. Basically, a cyanobacterium was engulfed by a eukaryote, and that became the chloroplast. And Archaea plastida, the group, that they inherited that chloroplast, the primary endosymbiont. But then other groups of eukaryotes, they got the chloroplast via secondary or higher order endosymbiosis, and these include the euglenids, the chlorarachniophytes. You also have various groups of chromists, and chromists it's not a clade. It's just a group that it's just a name that refers to various groups which received chloroplasts that are ultimately derived from red algae via serial transfers. And these chromists include the cryptomonads, the haptophytes. The haptophytes also include the famous coccolithophores, which are like these coccolithophores are the fossils of coccolithophores. They form these white chalk deposits, like the famous cliffs of Dover. The white cliffs of Dover. They are just mountains of coccolithophore fossils, basically. You also have mesozoans, and mesozoans include the dinoflagellates and also the apicomplexans. Apicomplexans include the parasite Plasmodium that causes malaria. And apicomplexans are an example of a group that lost the ability to do photosynthesis, but Plasmodium parasites still contain plastids, but the plastids are no longer able to perform photosynthesis. And lastly, you also have the group called ochrophytes. These ochrophytes include the diatoms and brown algae like kelp as well. And again, if you want to know more about these groups, we have discussed these groups in detail in the first chapter of photosynthesis. Our next slide. So first we go over the group that have the primary endosymbionts, the Archaea plastida, which are the chlocophytes, the rhodophytes are the red algae, and the last group is Viridae plantae, which are the green algae and land plants. Next slide. So as I mentioned before, in land plants, you have this particular supercomplex of photosystem 1 with these light harvesting complexes. It turns out that when you compare the land plants and the green algae cousins of land plants, green algae tends to have more of these light harvesting complexes, probably because green algae tend to be living in water, and with more light harvesting complexes, you probably need to compensate for being in water. Land plant, of course, when you're on land, you're at greater risk of overexposure to sunlight. Yeah, makes basically sense. Also, these super complexes are also very similar in the euglenids and chlorarachniophytes, because of course the euglenids and chlorarachniophytes, they have obtained their chloroplast by engulfing green algae, and that makes sense. They have the similar super complexes. All right, next slide. Also, before I also forgot to mention that these light harvesting complexes have chlorophyll A and chlorophyll B as well. There's a major feature of these light harvesting complexes that are found in green algae and land plants. But in rhodophytes, you have something a bit different. They also have light harvesting complexes which are related to those found in the green algae and Viridae plantae, green algae and land plants. But in rhodophytes, the light harvesting complexes only bind to chlorophyll A, and there is also no xanthophyll cycle. So as I mentioned before, the xanthophyll cycle is a way to dissipate excess energy, but rhodophytes lack this ability to dissipate this excess energy of light. And it's probably one reason why rhodophytes are not very common on land. Like there is one interesting group of rhodophytes, and I give one example all the way on the top left. You have this group which lives in terrestrial hot springs at a very acidic temperature, like a battery acid almost, and also at very high temperatures. These are thermophiles. And also, interestingly, rhodophytes also have a very different light harvesting complex called the phycobilosomes, and they consist of phycobilla proteins, and they don't have chlorophyll in them. They have a different group of pigments called phycobilins or phycobilins. I'm not sure exactly how you pronounce these, phycobilins or phycobilins. It's also interesting to mention that you also produce, you don't produce phycobilins, but you do produce similar compounds called bilins, which are breakdown products of heme metabolism. Basically, when your red blood cells run down, they degrade. your liver, I think it is your liver that detoxifies the excess uh, hemoglobins found in the red blood cells. Maybe you're more familiar about the Jackson? I'm not exactly sure if I'm right about that.
I'm also not sure. I mean, that would make sense to me, but I'm not
Yeah,
positive.
I think it's the liver and the breakdown products of the hemoglobin that goes into the gallbladder, which produces these biles, and that breakdown product is first green colored, hence why bile is colored green due to these pigments of bilins, and eventually the digestive system modifies these bilins to become a brown pigment, and that is why your feces are brown color usually. And if something goes wrong in digestion, then the pigments remain yellow, and that is why sometimes if you have some indigestion, your feces become yellow. So one way to explain this color, the color pigments. But yeah, but these in the rhodophytes, in the phycobilosomes, they have a different set of pigments called phycobilins specifically, and they have very different properties. Like rhodophytes in particular, they have a very high amount of a subset of phycobilosome proteins called phycoerythrin, and that erythrin is basically refers to the fact that these pigments are red colored. Hence, why most rhodophytes are colored red. And these phycobilosomes are also they are very big. They are very efficient in absorbing light that penetrates deep down in the water column. And you can see one example on the top middle right there. This species Asparagopsis. That species is found very deep, 40 meters down in marine environments. It's also an extremely invasive species in the Mediterranean as well. And you can see that it's a very dark red colored. That helps it absorb the red light that penetrates deep down in the water column. Also, interestingly, the species that I mentioned before on the top left that lives in terrestrial hot springs, that one lost the phycoerythrin proteins. And the reason why is because in the terrestrial environment, they don't have to absorb the red light. In fact, when you're living in terrestrial environments, absorbing too much light can be detrimental, and that is why these terrestrial species have lost the phycoerythrin. Yeah. All right. Next slide. And then the third group of the third group of Archaeoplastida, these are the glaucophytes. They are very as of now. I think they are. I think they probably are more diverse than we know. But as of now, we are only aware of a couple of dozen species, 13 to 26, and they only reproduce asexually. There's no sexual reproduction that we know of among them. And they also have phycobilosomes, but they also have lost phycoerythrin as well, like one of the subgroups of rhodophytes. And of course, as you may guess, these glaucophytes are also exclusively terrestrial, which is probably the reason why they lost the phycoerythrin proteins, which I have abbreviated as PE, that is phycoerythrin. Yeah. And that's also the reason, and since they lost the red pigment, that is also why they appear to be a bluish green color due to the loss of the pigment. Also, interestingly, I have, as you may, I have given schematics of these photosystem 1 and photosystem 2 supercomplexes on the images right there. In land plants, the photosystem 2, they form a dimer, a structure consisting of two PS2 systems. Photosystem 1 is just a monomer. It only occurs as a copy of one as a structure. But in glaucophytes, the PS1 system, they form a tetramer, like a series of 4 copies of the same complex. It's a very interesting difference in glaucophytes. Next slide. And then we move on to the cyanobacteria, which is of course the origin of the chloroplast found in eukaryotes. They also have the phycobilisomes. So rhodophytes and glaucophytes, they have inherited the phycobilisomes from the cyanobacteria, but the green algae and land plants, they lost the phycobilisome altogether at some point. And this is also probably the reason why that land plants were able to conquer the land, because phycobilisome is not very compatible in a terrestrial ecosystem. Because I will mention exactly why that is the case later on as well. Let's see here what there are. Oh yeah, there are also other light harvesting complexes found in cyanobacteria. You have also a reduced version of phycobilisomes called the CPCL complex, which associates with both the PS2 and the PS1 system. Also in cyanobacteria, the PS1 system forms a tetramer, a series of 3 copies of the PS1 system, as you can see on the top end of the slide. There are also additional photo harvesting systems. One of them is the ISIA proteins, which forms a ring around the trimer of PS1. And you also have another. First, before I move on, the ISIA light harvesting system. The cyanobacteria only make this complex when they are under iron poor condition. So when there is iron available, this ring of ISIA proteins, they don't form around PS1 system. But when the iron nutrient is very poor, then the cyanobacteria expresses the protein, and then they surround the PS1 system. Another light harvesting system in cyanobacteria is the PCB light harvesting proteins, which do bind. This is also very peculiar. The PCB is a protein that binds to chlorophyll A and B. However, it is unrelated to the light harvesting complexes found in green algae and land plants. And again, remember, the light harvesting complexes in land plants, they bind to chlorophyll A and B. But in this case, some cyanobacteria have evolved an enzyme or a protein independently that also binds to both chlorophyll A and B. Next slide. Also to give you an explanation, the PCB protein and also the ISIA protein that I mentioned before, these light harvesting proteins found in cyanobacteria, they appear to have been they are duplicated versions of the CP43 protein. And this CP43 protein is one of the subunits of photosystem 2. So basically, it's a copy of a subunit of photosystem 2, which evolved into light harvesting complexes in cyanobacteria. And there are a few citations that I've given to support this, in case somebody wants to object. There are the citations. You can look at them if you want to. All right, next slide. So here's the comparison between the super complex found in land plants, which with the light harvesting complexes that bind onto chlorophyll A and B. And then on the right-hand side in cyanobacteria, the phycobilisome is the most common, I do believe the most common light harvesting complex found in cyanobacteria, which is humongous. Like it's much bigger compared to the light harvesting complex found in land plants. And that's not because bigger is better. As I mentioned, sometimes bigger is not better because on land you don't want to absorb too much light because that can overload the photosynthesis system. Next slide. So another way to compare these light harvesting, one implication of these light harvesting complexes is the effect they have on the structure of the the thylakoids. So again, remember, within the chloroplast and also within cyanobacteria, you have these membrane-bound structure called thylakoids, which consist of grana. In land plants and green algae, the Viveridae plantae, they are stacked, as you can see on the right-hand side of the image below. But in red algae and also glaucophytes, they have these big phycobilosomes, and those big phycobilosomes, they prevent these grana to become stacked. So they don't have true grana in their chloroplast. There's one big difference between the green algae, the green algae and land plants, and then compared to the red algae and glaucophytes. The fact that land plants and green algae have these stacked grana also allows for an optimal ratio between the PS1 and the PS2 system that is more suitable to fix CO2. And this can meet the high demand for biomass to produce cell walls and lignin. So this difference is probably one of the reasons why land plants conquered land, because they are able to basically the grana structure in Viveridae plantae facilitates an optimal ratio of PS1 to PS2 such that they are able to fix carbon more efficiently to produce lignin. And again, as you may have known, lignin is the structure that produces wood, trees of wood, basically. Yeah. All right. Next slide. Also, this is also an interesting story about the evolution of the phycobilosomes within the chloroplasts. So, as I mentioned before, that while land plants have stacked grana are not essential to photosynthesis because, again, red algae and glaucophytes, they don't have stacked grana, but they can still perform photosynthesis nonetheless. But there's another interesting fact is that thylakoids are also not necessary for photosynthesis. There is in cyanobacteria, the cyanobacteria that perform photosynthesis basically split into two ancient groups. One major group have thylakoids with basically internal membrane structures, the thylakoids. But you have another group, they are called the Gliobacteria. They don't have thylakoids. They perform photosynthesis on their outer membrane, basically. And in one of the papers that I cite here, they include this image, and they just describe one of these cyanobacteria that lack thylakoids, and they also have a very differently structured phycobilosomes, like a phycobilosome without rod structures. So, like most of the typical phycobilosomes, they have these rods structures on them, but in some of the cyanobacteria without thylakoids, they lack the rod structure of they have more simple phycobilosomes compared. And the authors of this paper proposes this scenario for the evolution of phycobilosomes. But unfortunately, we don't have time to go into detail about how phycobilosomes evolved step by step. This is just I think it's a very neat image that just summarizes the overall hypothesis of how the phycobilosome evolved and became more complex in these two groups of ancient cyanobacteria. And it's also a good illustration that to counter one point of irreducible of the suggestion that photosynthesis is irreducibly complex, the fact that thylakoids are not necessary for photosynthesis. You can have a cyanobacteria without thylakoids still
Nesslig,
being able to produce. Oh,
can
yes?
I barge in for just 1 second? BBS666 for 199 euros. Thank you so much. That was all I had to say. Thank you.
Someone from Europe apparently with the euro. Thank you for the donation. All right. Next slide. All right. Also, I've also found a few papers discussing how the thylakoid was formed. So the traditional hypothesis suggested that the thylakoid originated by invaginations of the plasma membrane. So remember that the bacteria have an outer membrane called the plasma membrane. And basically, the traditional hypothesis suggests that this plasma membrane invaginated on the inside, and that invagination budded off, producing the thylakoids, becoming an internal membrane structure, the thylakoids. But a new hypothesis suggests that the thylakoids originated from basically a phase transition involving non-vesicular glycolipids. Yeah, this involves a very complicated physics, phase transitions. So it's this is in the realm of physics that I'm not very much familiar with. So what they suggest is that if I remember correctly, there are two types of these lipids. One forms a hexagonal structure, and another forms these laminar structures. And what they suggest is that at one point, if the ratio changes up to a certain state, then you have a phase transition that allows the formation of these thylakoids. And that's how they suggest the thylakoids originate. And again, more details can be found in the papers that I cite. And I'm not too super familiar with this process myself. But it's also interesting to note that thylakoids, what thylakoids do is they increase the surface area for photosynthesis by 10-fold. So this is also, again, the reason why, again, as I mentioned in the previous slide, there are two groups of cyanobacteria, those with thylakoids and those without. And as you may have guessed, the cyanobacteria with thylakoids, they are much more common because the thylakoids, they enable more efficient photosynthesis because the surface area, photosynthesis is dependent on the surface area of these membranes as well. Yeah. All right, next slide. Oh, now we move on to other groups of eukaryotes, the chromists, the so-called chromists, and we go over each group one by one. Next slide. So again, as I mentioned before, the chromists, they inherited their chloroplast from the red algae. And the first group is the cryptomonads. They also have the same light harvesting complexes that red algae, the rhodophytes, have, but they have there are a few changes as well. In the cryptomonads, these light harvesting complexes, in them, they are called ACP proteins, but they are still related to the light harvesting complexes of rhodophytes. But in the cryptomonads, they contain more pigments compared to the rhodophytes. They contain alloxanthin. That's a carotenoid. They contain chlorophyll A, but they also contain a different pigment called chlorophyll C2. That is unique to the chromists, or in this case, the cryptomonads. That is not found in the rhodophytes. They also have phycobilosome or phycobiliproteins as well, but these phycobiliproteins, they don't form phycobilosomes. What they do is they form these hexamers or trimers that are found within the lumen, like free floating within the lumen. So it's almost like the phycobilosome just broke, and little bits are floating around within the lumen side of the cryptomonads. So they have proteins related to the phycobilosome, but they don't form the phycobilosome structure anymore. But they still absorb, they still harvest light for photosynthesis nonetheless. So this is one example of, like, again, evolution is often mistakenly taken to be a progressive, stepwise evolution to become increasingly more complex and better over time. But sometimes things can evolve to become simpler, like in this case, the phycobilosome was lost, but some of the bits stuck around, and now they are simpler in structure. All right, next slide. Now we move on to the haptophytes. Again, in this case, the haptophytes also include the coccolithophores that I mentioned before. They have these calciferous, how do you call them again? The shells. They are made of calcium carbonate, yes? If I remember correctly. Jackson?
Yes. Yeah,
Yeah, they
the
are
shells
made,
are
yeah.
made of calcium carbonate. Yeah,
Yes, they
sounds
are.
right.
Yeah, and you can see one example on the bottom in the middle right there. Yeah, and also the structure that I've crudely made little icons on the slide to basically give you a rough idea of how these super complexes look like, and they are based on the structures from these two. Uh, yeah, these two species or genera. So photosystem one is based on the Coccolithophore emiliana or emiliania, yeah, emiliania, and also photosystem one. The structure for photosystem one is based on the species on the left hand side, right there. Like there's also maybe a thing that I can make note of. Like we do know some, like we can have crystal structures or 3-dimensional structures of these photosystem super complexes, but we don't know of every single species, of course, because doing that work is insane. There are too many species to analyze. So what they do is they just pick examples or they pick model species and they take that to be a representative of what is found within one group. But sometimes there is still more variety among the same group, of course. But these are just one example of a structure found in the haptophytes that I'm showing right now. So in this case, as you can see in the photosystem 1, they have an extreme amount of light harvesting complexes. And some of the light harvesting complexes are also associate with photosystem 2 as a dimer form. So that is also interesting to know. So in the land plants, some of the light harvesting complexes are trimers. They are like groups of 3 polypeptides. But in the case of haptophytes, some of the light harvesting complexes surrounding the PS2 system, they are dimers, copies of 2 polypeptides. And in the case of the photosystem 1, it's surrounded by a huge amount of light harvesting proteins. And these are also unique by they bind to another unique carotenoid called fucoxanthin. They also bind to chlorophyll A as well as chlorophyll C1, C2. And some of the haptophytes also have another chlorophyll called C3 as well in their light harvesting complexes. And they have also uniquely they have an alternative xanthophyll cycle. I mentioned the xanthophyll cycle in land plants and also green algae, but the haptophytes have a different version of the xanthophyll cycle with involving different carotenoids, and this allows them to dissipate excess light energy as well. Next slide. So there's a paper that's discussing the evolution of these super complexes and light harvesting complexes, and it turns out that within the coccolithophores they just have a huge number of these light harvesting complexes compared to other groups of algae as well. And my guess is, like as I mentioned before, coccolithophores have these calcium carbonate shells, and my suspicion is this is my suspicion. I haven't seen anything found in the research specifically, but my guess is that the reason why they have these large number of light harvesting complexes is because it's a way to compensate for the fact that they have this calcium carbonate shell that limits the amount of sunlight that can get through the cell, of course. Yeah. All right, next slide. So now we move on to the mesozoans. And the mesozoans, there is not a lot information about the mesozoans. Like again, we know about the dinoflagellates as well. And in dinoflagellates, I could not find an example of a known structure for photosystem 2, but we do know about a unique light harvesting complex called the PCP, or the full name is pyridine chlorophyll protein complex. And this PCP light harvesting complex, it is dissolved within the lumen. It is free floating in the lumen, and it is unique to the dinoflagellates within a subset or a subgroup of mesozoans. You also have I think I misspelled. Like in the text on the top right, I misspelled symbiodinium. Symbiodinium is a genus that is a common symbiont of corals, right? Yeah, corals often have these symbiodinium as their symbionts that allow chlorophyll that allow corals to grow on sunlight.
Yeah.
By having this symbiotic relationship. And we do know the PS1 structure from symbiodinium as well. And the structure looks like this. It also contains the same light harvesting complexes related to the ones that that's also I forgot to mention before. Like I color coded the light harvesting complexes based on their homology. Every single one that is colored pink right here, they are all related to each other. They are homologous. And related to the light harvesting complexes found in the rhodophytes all the way on top. Yeah. Mm-hmm. All right. Also, one last thing to note is that the mesozoans, they also have light harvesting complexes that bind to fucoxanthin, chlorophyll A, also chlorophyll C1, C2, and sometimes C3 as well. And they also have, similar to the haptophytes, they also have the alternative xanthophyll cycle that allows them to dissipate excess energy. Next slide. And now we move on to the last group of chromist called the ochrophytes. This group contains the kelp, the brown algae, and the diatoms. I give you one example of diatom over there. And from this diatom was used to figure out how the light harvesting complexes look like as a three-dimensional structure. In this case, the diatoms also have an excessive amount of light harvesting complexes. And again, similar to the coccolithophores recently, diatoms are also armored, but in this case they have silica armor. They are surrounded by glass, almost glass. Yeah. And this is probably the reason why they have also excessive amounts of light harvesting complexes surrounding photosystem 1 and also photosystem 2. And in photosystem 2, some of these light harvesting complexes are tetramers, groups of 4 polypeptides. Mm-hmm. All right, next slide. So as I mentioned before, all these light harvesting complexes are related to each other, and they're also related to the light harvesting complexes found in the green algae and the land plants. And here is a few papers that show the evolution of these light harvesting complexes. So they are hypothesized to have evolved from a one helix protein, similar to the what is called the HLIP/OHP1 protein found in Photosynthetic and also a few of them are found in eukaryotes as well. This is a one helix protein, and the one helix protein is noted by a 1 bar. But then over time, the one helix protein evolved into a 2 helix protein, and then later on into a 3 helix protein by either gene fusion or helix domain duplications. And the 3 helix proteins, they are the light harvesting complexes. Yeah. Also, at the very beginning of the presentation, I mentioned a regulatory protein, the PSBS. This is a regulatory protein that regulates the process of light energy dissipation. the PSPS is also a part of this superfamily of light harvesting complexes as well. So yeah, very interesting. Is there any questions about in the live chat so far? I'm not sure. I'm not paying much attention to the live chat right now.
Not currently.
All right. I hope I'm being entertaining to everybody here. All right. Next slide.
I'm enjoying it.
All right. So now we finished the story of eukaryotes. Now we move on to the bacteria. Next slide. So as I mentioned in the first chapter about the sun eaters, in bacteria, as of now we know about 9 bacterial phyla that include members that can perform some form of photosynthesis or some form of phototrophy. So again, this is some background that I need to mention again. Like photosynthesis is not always photosynthesis. Sometimes these bacteria they can absorb light energy and use the light energy to make ATP, but they are unable to use the light energy to fix carbon dioxide. So they are photoheterotrophs. One example of that is the all the way on the top of the list, the acidobacteria. They are photoheterotrophs. They are able to capture sunlight and use the light to make ATP, but they are not able to perform carbon fixation with that. Also, one thing to note is you in addition to bacteria, you also have the archaea, and some of them are also phototrophs, but they use a different pigment. They use retina pigments or retinol-based pigments. But bacterial phototrophs, they use chlorine-based pigments, like chlorophyll. Also, while we know of 9 bacterial phyla that have phototrophic members, we only know the details for 6 of these. Like as I mentioned, like in the table on the left, I notated with purple which phyla are only recent discoveries. So there's one phyla that was discovered in 2014 to be photoheterotrophic, and also a few others. So more recently, one in 2018 and one in 2023, very recently, was found to have potentially phototrophic members. But as of now, we only know the details for the 6 other phyla. Next slide. So this is a basic description of how photosynthesis looks like in cyanobacteria. You have photosystem 2, and on top of that you have the phycobilazome, of course. Then you have the cytochrome P6F complex in the middle, and then you have the PS1 system, and both are used to power the Calvin cycle to fix carbon dioxide into biomass. Also, one thing to note is that in cyanobacteria you have 2 types of photosystems. Again, photosystem 1 and photosystem 2. Photosystem 1 is the iron sulfur reducing system, and photosystem 2 is a quinone reducing system. Next slide. But in the other phyla or bacteria, they only have 1 of each of these types. So two of them have the quinone type, or in that case, like in cyanobacteria, they are referred to as PS1 and PS2, but in the other bacteria, they are called reaction centers, or RC1 and RC2. RC1, as the name suggests, RC1 is like PS1, and RC2 is like PS2 in this case. I'm not sure exactly why they chose to just call them with different names. I would have, if I could travel back in time, I would just say, like, just use the same name for all of them. Just say PS1 in all of them and PS2 in all of them. But in any case, it's about, yeah. There are also some structural differences between these systems. So the PS2, PS1, and RC1 are heterodimers, but the RC1 in non-cyanobacterial bacteria, the arsenal are homodimers, and this will be very important later on as we'll discuss the details of how these reaction centers have evolved. Next slide. So, as I mentioned before, chlorophyll is very important. Cyanobacteria uses chlorophyll as their redox active, as their photoactive redox centers. So, in photosystem 2, you have the P680 complex, which consists of chlorophyll. In PS1, you have the P700 complex, also chlorophyll A. There are some cyanobacteria that are adapted to far red light, which replaces chlorophyll A with chlorophyll D. And when it replaces chlorophyll A with D, it becomes the P700 becomes P740. And the reason why is because now the wavelength that it absorbs shifts to longer wavelengths. So instead of absorbing P700, it now absorbs higher wavelengths of light. Hence why it's far red light adapted. There are also other far red light adapted bacteria that live inside stromatolites, and they use chlorophyll F as the accessory pigments in addition to chlorophyll A. But in the non-cyanobacteria phototrophs, they use bacteriochlorophylls. So the heliobacteria, they use bacteriochlorophyll G. Acidobacteria and chlorobi, they use bacteriochlorophyll A. Also the purple bacteria for short, like proteobacteria, they are often called purple bacteria because they seem to be purple to us. Due to these pigments, and they have chlorophyll A. Also, chloroflexi also have chlorophyll A as well. And you can see here that these photoactive redox centers, they have the names P800, P840, P870, P865. They absorb very long wavelengths compared to the chlorophyll found in cyanobacteria. Also, interestingly, that some acidophilic bacteria, they replace the magnesium, like chlorophyll. Normally, chlorophyll and bacteriochlorophyll, they contain magnesium within the molecular structure, but sometimes in acidophilic bacteria, that magnesium is replaced with zinc. And the reason why is it seems to be that under very low pH, under very acidic condition, bacteriochlorophylls with zinc is more stable under these conditions. All right, next slide. So now you move on to the bacterial diversity of electron donors. So in cyanobacteria are the only ones that are oxygenic. They are the only ones to take electrons from H2O and then leave O2 behind as the waste product. Some cyanobacteria can also use hydrogen sulfide or H2S as the electron donors. And when they use that, they use sulfur as the waste product. Other bacteria, none of them can use water or H2O as the electron donors, but they have a wide variety of electron donors. They can use organic compounds as the electron donors. Some use sulfur, sulfides, sulfates, hydrogen gas, nitrites, ferrous iron. Some also use arsenite or arsenic as the electron donors. And I believe some of them, like there was a while ago, there was this news on where people found bacteria that live in these arsenic lakes. And some people claimed that these were life unrelated to other life. Do you remember that, Jackson?
Yes, I do remember that
Yeah.
claim. Yeah,
Yeah, so
another example of
Mhm.
hype for
Yes.
nonsense in the media.
Yeah, so these people claimed that, like in DNA, you have like phosphorus is one of the components of DNA, and these people claimed that these bacteria have arsenic in place of phosphorus in their DNA. And based on that, they claimed that these bacteria were unrelated to all other life, but that turns out to be completely false. Yeah. I have to get some more water. I'll be right back in a few minutes, or a few seconds, actually.
And in the meantime, I'll distract everyone with my juggling. Can we ever have a scientific discovery without people saying it's aliens? The answer may surprise you. It's no. Yeah, that's an example. Aaron likes to bring up when talking about how bad like pop sci is at relaying science. Or what was it? The one with Ida, the Darwinius Mastele. They said, "Oh, she's the ancestor of humans." Or there was another example, something ridiculous. Oh, I think Peter, wasn't there a paper? There was some paper that came out about. I think it was just something about like the dietary habits of Neanderthals, but a pop sci article concluded based on this paper that Neanderthals were cannibals, which was not what the original paper had said. Do you remember that, Peter?
I do recall a paper about that, but I honestly don't remember the details anymore.
I don't remember the details of the original paper. I just remember people being like, "What the heck? This is not what the paper says."
Yeah,
because they were saying it was cannibal Neanderthal.
Yeah, I have seen these like red-eyed, black-furred Neanderthals, right? Right? Very scary Neanderthals that are like hunting humans, basically.
Which is funny because apparently, so there was a paper that came out like a week or so ago, or a couple weeks, that showed introgression between Neanderthals and humans was very sex specific. It was mostly female humans mating with male Neanderthals. So
yes,
do with that information what you will.
Well,
I think it's also like we discussed this in one of the ancestors tales a while ago. Yeah.
Also, I'm kind of impressed how good you can juggle while talking science.
I know, and my hands are different. Looks like different colors.
No. Just one's near the stage light, and the other is
just in the shadow.
Yeah, it's a little in the shadow.
Gotcha.
That's what you get when you go on YouTube and you don't have professional lighting, and what happens. Okay.
All right, we can move on to the next slide.
Okay.
Okay. So Jackson should stop juggling.
Yeah, I think I'm done juggling.
Next slide.
Yes. Also, there's also some diversity in the quinone acceptors. So I remember that in cyanobacteria, you have the cytochrome B6F complex. But in other bacteria, as like some of them also use the B6F complex. In heliobacteria, they use that. But chlorobi and proteobacteria, they use the cytochrome BC1, the same cytochrome that we have in our mitochondria, basically. And you also have two other groups, the acidobacteria and the chloroflexi. They use an alternative complex called AC3, which seems to be, as far as I can tell, this seems to be not related to the other ones, but I'm not sure about it exactly. Also, cyanobacteria, they use a quinone called plastoquinone, or PQ, but heliobacteria, chlorobi, and also the other ones as well, they use a different quinone. I think it's called MENA quinone, MQ. I abbreviated it as MQ, but I think the full name is menaquinone, but I'm not sure exactly. Also, proteobacteria, they use MQ or UQ, and UQ that refers to ubiquinone, and that is the same. And we also use ubiquinone as the quinone in our mitochondria as well. Yeah. And you may have guessed that Proteobacteria, that is the same phylum from where our mitochondria was derived from. Because our mitochondria are also endosymbionts as well. And that is the reason why mitochondria has the same proteins, the cytochrome BC1 and also the ubiquinone as well. All right, next slide. Oh, can you go back for 1 minute? Sorry about that. I think I forgot something. Yes. Yeah, the one other note, the heme-based soluble cytochrome C are often used instead of the copper-based plastocyanin. So I mentioned before that the electron from in cyanobacteria and also the land plants as well, the electron from the electron transfer from B6F to photosystem 1 that is mediated by plastocyanin that moves through the lumen. But in other bacteria, instead of plastocyanin, the cytochrome C is used instead of the copper-based plastocyanin. Yeah. All right. Yeah. Oh, yeah. And that's my final note on this slide. Yes, you can move on to the next slide. Sorry. All right. So there was also so we have mentioned that some of the diversity of reaction centers, some of the diversity of the cytochromes as well. But there is also an incredible diversity regarding the light harvesting systems within bacteria as well. So again, you have the phacobilisomes found in cyanobacteria and also other light harvesting complexes found in cyanobacteria. Within proteobacteria, they rely on a different light harvesting system called LH1 and LH2. And you can see some of the structures proposed for these proteins on the images right there. They are also very interesting as well. But unfortunately, not much is known about these systems and where they exactly where they come from. Next slide. And the main reason why is because these bacteria are very understudied. Like the things I'm showing you right now is probably just the tip of the iceberg, the tip of the proverbial iceberg, as they say, of what is really known out, what is really out there.
Well, if it's not big for like agriculture or medicine, then who cares? So
right. Mm-hmm. Yeah, like half of all paleontology studies is dinosaurs, and even though it's only like a tiny part of the Phanerozoic.
Right.
Right. Okay, so continue. This was another light harvesting system found in non-cyanobacterial bacteria. You have these 3 groups of these non-cyanobacterial bacteria. They have these huge chlorosomes. They are by far the largest light harvesting system that we know of. And they are basically like membrane bound ink bottles, just chock full of bacterial chlorophylls and probably also many other pigments. It's just filled with these pigments that aggregate without a protein scaffold. It's just a blob filled with pigments, and they are attached on top of these reaction centers, as you can see on the image right there. Yeah, and this is probably also these bacteria that it possesses these chlorosomes. As you may have guessed, they are found in environments that are very dark, like they are found in soils, and some of them, as I will mention later, they are also found in deep sea hydrothermal vents as well. And you may not guess that they are found even in those environments as well, but they apparently are. Next slide. So one other thing to also notice that in one group is called the heliobacteria. Is the one all the way on top of the image on the left top side right there, they don't appear to have a peripheral light harvesting system. The light harvesting capacity is inherent to the RC1 system that they have. So if we can make another note against irreducible complexity, light harvesting systems are also not necessary for photosynthesis. You can some of the light harvesting capacity of inherent to the reaction center, it can be sufficient to perform some form of phototrophy in the case of heliobacteria. Yeah. All right, next slide. And now we move. A final note is on the carbon fixation mechanism. So again, cyanobacteria, they use the Calvin cycle to fix carbon. There is also the in proteobacteria, also called propylbacteria. Again, they also use the Calvin cycle to fix carbon. But in green sulfur bacteria, they use the reverse TCA cycle. The TCA cycle refers to the tricarboxylic acid cycle. It's more commonly known as the citric acid cycle. The same citric acid cycle that occurs within our mitochondria, except green sulfur bacteria drive this cycle in reverse. So instead of producing CO2, they take CO2 and turn it back into biomass in the opposite direction compared to our citric acid cycle. You also have chloroflexi, members of the chloroflexi group. They don't use the Calvin cycle. Chloroflexi, they use either the Calvin cycle or they use a very different cycle called the 3-hydroxypropionate cycle. This is a very unfamiliar cycle. There are some similarities to the citric acid cycle, but in some ways the details are very different in some other ways. Yeah. All right. So, also two groups are heterotrophic. They don't fix carbon. They don't have a carbon fixation cycle. So in this case, the electron transfer that occurs within them is cyclical. There is no final electron acceptor in their case. Next slide. So yeah, so you have a system like this in cyanobacteria. Next slide. But another counterpoint to irreducible complexity is that you don't have to fix carbon. This system can still function in a simpler way by just doing cyclic electron flow. And cyanobacteria are also able to perform this electron flow by using another enzyme called NDH1, which is homologous to our complex 1 in our mitochondria as well. It's also interesting to note is that, like in cyanobacteria, the electrons flow back via NADPH, but in land plants have lost part of the NDH1 complex, and this means that electrons only flow back via ferredoxin from this system. So there's a slightly different way how the cyclic electron flow works in cyanobacteria compared to the chloroplast found in land plants. Next slide. Yeah, and then the cyclic electron flow allows them to make ATP, but it does not allow them to fix carbon. But it's still very useful to make ATP regardless. So again, main points against irreducible complexity so far. Photosynthesis is highly modular. Different modules can still be functional, even in the absence of some common components. Like, as I mentioned before, you don't have to have stacked grana. There are organisms like red algae that don't have stacked grana. There are also cyanobacteria that don't have thylakoids. There are also bacteria like the heliobacteria that lack like harvesting proteins and also no carbon fixation pathways. So there are several different ways in how you can reduce photosynthesis and still have it functional nonetheless. But one counterpoint that can be made is that don't you need carbon fixation from photosynthesis to have to maintain an ecosystem? Well, that question will be asked in the next few slides. Next slide. So now we move on to what life does in the dark. Next slide. So, as I mentioned before, within a lifetime, like before the 1970s, every scientist thought that photosynthesis was necessary for life. Without photosynthesis, the ecosystem will just collapse and life will cease to exist. But then you have this little guy called the Alvin Explorer, and they went down to spreading centers near the Galapagos Islands as well. And when they descended, they were just following heat signature. So they knew that they were going through they were heading towards deep sea hydrothermal vents, but they believed that no life would be found all the way down there, away from light, and there's no possibility for photosynthesis deeper down at that level. So they were very shocked to find that they found a very thriving ecosystem. It's almost like discovering alien life. Sorry, Jackson. They almost, of course, this is not alien life, of course, but it was really almost like landing on the moon and then finding life.
Yeah, it is pretty crazy.
To them.
I mean, it's, in some ways, I think it's kind of poetic. I think it's poetic that like the Galapagos was both the place where Darwin was getting his ideas about natural selection, and also the place where we discovered hydrothermal vents that aided our understanding of the origin of life, and
Exactly.
radically changed our understanding of biology. So yeah, I think that's very poetic.
Right.
Also, Nesslig, I think you may have irritated Smitty now. Just look at the third comment from the bottom.
Is ubiquinone the chemical opposite of
No.
unobtainium? No.
No, Smitty's comment.
Can we ever have
scientific discovery without people saying it's aliens? Smitty called it.
Sorry, Smitty. Yeah.
He called it.
Yeah, well, luckily, nobody suggested that these deep sea ecosystems were aliens. In fact, so no worries, Smitty. There's no need to raise alarms in this case. All right, so also among the most abundant life forms found in this dark ecosystem was the giant tube worms called Riftia, and as you may guess, Riftia is named after the rifts within these the deep sea rifts that are the cause of the hydrothermal vent systems. Also, interestingly, Riftia, the giant tube-worms, are some of the fastest growing organisms that we currently know of. Like in terms of building biomass per unit of time, they are the fastest growing organisms. Next slide.
You're telling me there are giant, fast-growing worms? I don't like that.
You're right. Yeah. Also, the adult giant tube-worms. Also, interestingly, they like when they are I'm not sure I think they are called larvae at the young stage. I think so. When they are young larvae, these tube-worms do have a mouth and a gut and an anus. But when they mature into adults, the mouth, guts, and anuses are completely lost. What they do is they have this plume structure which absorb CO2, oxygen, and also hydrogen sulfide. And these plumes are also colored red because they have hemoglobin, a form of hemoglobin. It's also interesting to mention is our hemoglobin is a 4-unit structure. It contains 2 alpha polypeptides and 2 beta polypeptides. But these giant tube-worms, they have gigantic globins. Like one version, the V1 type, consists of 144 globin units. And another type, V2 and C1, they are They consist of 24 globin units. And this unique globin units, their special hemoglobin is able to bind onto oxygen and hydrogen sulfide at the same time. Like our hemoglobin is not able to do this. Which is why hydrogen sulfide will poison us very quickly. But these two worms are not bothered by this because they have these special hemoglobins that can bind onto oxygen and hydrogen sulfide at the same time. Yeah. Also, it's also interesting. These different versions of hemoglobin are found in different tissues. I forget which tissues. I think the V1 and the V2 are found in the vascular system, and the C1 is found in the coelom, if I remember correctly, but I'm not exactly sure about this. So can I still hear you? I have no audio anymore, I think.
We can still hear you.
Oh, sorry. Yeah. Now I still hear you. Yes. The battery of my earphones went down, so I think I just replaced it. But so now I can hear you. Yes.
Oh, you sound fine to us.
All right. Now it was on my end. My earphones, like one of my earphones battery died, so I had to replace it. And now I can hear you again. All right. Okay, moving on. Like even though they lack a mouth, gut, and anus, they do have an organ called the trophosome that contains symbiotic bacteria, and these symbiotic bacteria they use oxygen to oxidize hydrogen sulfide, producing sulfur as a waste product. And this the energy obtained from oxidizing hydrogen sulfide that powers the Calvin cycle with these cyanobacteria. So they are able to fix CO2 without using sunlight. Next slide. And there are also organisms that don't depend on organic carbon nor oxygen produced by, like as I mentioned before, even though the tube worms can grow without sunlight, they do use oxygen, and oxygen is still dependent on the photosynthesis that occurs on the surface. But there is life that is completely independent from photosynthesis. They don't even need the oxygen produced by photosynthesis on the surface. They solely rely on the electron donors and electron acceptors provided by the geochemistry. Some of these include the acetogens and methanogens that use hydrogen or hydrogen gas as the electron donor. And this hydrogen gas is produced by a geochemical process called serpentinization. And they use CO2 as the electron acceptor in this process. So in other words, they fix carbon via the linear acetyl-CoA pathway that releases a net amount of energy. So as I mentioned before, that in photosynthesis, the electron transport and carbon fixation, they are almost like separate processes. One makes energy, and that energy is used to fix carbon. But in this case, the carbon fixation process and the energy release process are the same. It's really incredible. They fix carbon to release energy. So as somebody else puts it in the quote that I gave it on the bottom left right there, they are likely to gain energy in the process. The result is a geochemically provided benthic boondoggle in which microbes are given a free lunch that they are paid to eat. So this is very different from the photosynthesis. Photosynthesis is an uphill process. You need to use sunlight to fix carbon into biomass. But in this environment, without oxygen, without sunlight, the carbon fixation from using hydrogen gas is a downhill process that releases energy. Next slide. So here is the acetyl-CoA pathway in more detail. The acetyl-CoA pathway is likely one of, if not the most ancient, one of the most ancient biochemical pathway, and it is shared by archaea and bacteria. Bacteria that uses this pathway are called acetogens, and archaea that uses this pathway are called methan. Oh, sorry. Hold on. Am I correct in this? Let's see here. Acetogens. Maybe I made a mistake here. Acetogens are. Let's see here. Oh yes, I got this. I got this wrong on the slide. On the images I say acetogens are archaea. No, acetogens are bacteria, and methanogens are methanogens are archaea. So there's some mistakes on the slides here. So just to avoid any confusion, acetogens are bacteria that uses the left hand side of the image. There are some differences in detail of how they use the acetyl-CoA pathway, and methanogens, which are archaea, they use the right hand side of the image right there. And as the name suggests, the acetogens, they produce acetate as the waste product, and the methanogens they release methane as the waste product. But there is a common branch of the pathway in the middle right there. In the middle, that is common in both methanogens and acetogens. And also, coincidentally, they involve proteins that have these very peculiar iron, sulfur, nickel structures, which is very similar to the minerals found within hydrothermal vents. And this is also there's some very implications on the origin of life. Like some people like Nick Lane suggest that the mineral chemistry of hydrothermal vents led to the origin of life, and that mineral chemistry is preserved in these enzymes as these iron, sulfur, nickel cofactors or coenzymes. And I think you also are also familiar with Nick Lane, Y. Jackson.
Yeah, I read the Vital Question, and I mean the suggestion makes sense to me. I mean, you got like greigite and like pyrite and things like that are in the like alkaline hydrothermal vents that can we know catalyze a variety of organic reactions. So it makes sense to me.
Right. Also, other thing to note is that these bacteria, the acetogen bacteria and the methanogen archaea, they primarily use ferredoxin and another coenzyme or another molecule called the F420 as electron carrier. So in plants, in photosynthesis, the key electron carrier is NADPH, as I mentioned before. But in these bacteria and archaea, they don't use. Some of them use NADPH or NADH, but the primary electron carriers are ferredoxin and F420. Also, some of them lack cytochromes and quinones. Again, remember that in photosynthesis, the cytochrome, the main cytochrome is cytochrome B6F, and in us, the cytochrome is BC1, and they also use quinones that are associated with these cytochromes. But these bacteria and archaea, some of them lack cytochromes and quinones. The electron bifurcation process that is mediated by quinones, instead of using quinones, they use flavins as a means to perform electron bifurcation. Also, the redox centers are mostly iron sulfur clusters. Again, that's the same thing reminiscent of the mineral chemistry found in hydrothermal vents as well. There are also no chlorophylls are found in these bacteria. There are also no hemes. But interestingly, the methanogens do have a similar molecule, like chlorophylls and hemes. They are part of a group of chemicals called porphyrins, and porphyrins refers to the circular structure. As you can see the example right there, the coenzyme F430. That is an example of a porphyrin. You can see the circular structure right there. Chlorophylls are similar, and the hemes are also similar to that structure. No chlorophylls nor hemes are found in these bacteria or archaea, but the methanogens do have this coenzyme F430. And this coenzyme F430 is key for the production of methane in methanogens. And there's also there's also something to relate to the evolution of chlorophyll and hemes as well. The enzyme that makes this coenzyme F430, this enzyme is related to another enzyme that makes chlorophyll in cyanobacteria and also other bacteria, of course. And also, both enzymes are also related to another enzyme called nitrogenase. And you may be familiar with the nitrogenase as well, Jackson, or not? Oh, Jackson?
Sorry, I had to click back. Nitrogenase. I'm sure it's involved in the nitrogen cycle.
Yes, it's the enzyme that fixes nitrogen. Like you have nitrogen in the air as in the form of dinitrogen or N2. It is a very stable molecule. So there is not many life forms can fix nitrogen, but some bacteria are able to fix nitrogen by using the enzyme nitrogenase, and it's a homologous enzyme to the enzyme that produces this coenzyme F430, and also one enzyme is also used to make chlorophylls, or at least it is part of the pathway that makes chlorophylls as well. And this is something we will discuss in the evolution of chlorophyll in the future chapter as well. And but in the meantime, I don't know if you remember this, but you have like a long time ago you have debated Otangelo. I forget on what channel. I think it was on the non sequitur channel, right? Sorry to, or maybe on a different channel.
No, I debated Otangelo on MC Toon's channel.
All right. Yeah. So he brought up nitrogenase, I think.
He brings up a lot of stuff. That was a that I don't even remember much of what we talked about, because he just kind of runs off on various topics.
Yeah, but after that debate, I wrote a review of the debate on the Peaceful Science forum, and I discussed the nitrogenase on that forum. So if anybody is interested, you can go on the Peaceful Science forum and just look up nitrogenase Otangelo debate, Jackson Reed debate. You will probably find my essay on that forum on nitrogenase. All right. Also, one last thing that I have almost forgot to mention is that in these methanogens and archaea, the ATP synthase, like ATP in us, in our mitochondria, and also in the chloroplast of land plants, ATP synthase is driven by protons or H plus ions. But curiously, the ATP synthesis in these bacteria and archaea can also be driven by sodium as well. It's very interesting. And our next slide.
So on that point about the sodium, that's a point that Nick Lane makes in the Vital Question with regard to how sodium could have been one of the drivers of ATP synthase before it switched exclusively to protons in most groups.
Yes,
if I remember correctly. Yeah.
Also, like in these bacteria, you also have proteins that are related to complex 1. Complex 1 is one of the enzymes in our mitochondria. You also have, as I mentioned before, you also have a homolog of complex 1 in cyanobacteria and chloroplasts that is responsible for the cyclic electron flow. But in these bacteria, they also have homologs of complex 1. Some of them are in us and also in cyanobacteria and land plants. Complex 1 donates electrons, they use NADH or NADPH as the electron donor, and then they donate the electrons onto quinones. But in these bacteria that I just mentioned, these acetogens and methanogens, they have simpler forms of these proteins, and they use ferredoxin as the electron donor, and some of them use quinones as the acceptor. But some of the more simpler forms, they use ferredoxin as a donor and hydrogen ions as the acceptor, and that produces hydrogen gas. So they are able to, using these enzymes, they are able to use ferredoxin to produce as the electron donor. They use ferredoxin as the electron donor to make hydrogen gas, and that the energy released results in the pumping of or the translocation of hydrogen ions and sodium ions across the membrane. And that can power the ATP synthase too, because again, as I mentioned, the ATP synthase is powered by protons and sodium ions. And then further down, you have also a version of the version I just mentioned was the membrane-bound hydrogenase, the MBH. Next is the membrane-bound sulfur reductase, or MBS. That uses ferredoxin as the electron donor and then reduces sulfides to produce hydrogen sulfides. And again, this can both pump H plus and sodium plus ions as well to power ATP synthase. There's also another version as well, the MRP version, or multiple resistance and pH complexes. This is the simplest of all these complex 1 homologs, and they don't perform electron transfers. They are antiporters. What they do is they use I believe they use the proton gradient to power the translocation of sodium. So you can see in the, it may be difficult to see, but you can see the example of the protein right there. You see the movement of H plus ions from the top to bottom, and at the same time, sodium ions or Na plus ions are translocated from the top to the bottom in the opposite direction. They are antiporters. Yeah. And that is also probably also, I think it also relates to you mentioned Nick Lane, and Nick Lane suggests that an enzyme like this is responsible for the evolution of ATP synthase, such that they use natural, like these hydrothermal vents, they have natural pH gradients. They are like the inside of the hydrothermal vent is alkaline, it is high pH, and the outside is low pH. It contains an excess of protons. So what these bacteria can do is that you can use the environmental proton gradient to pump sodium, and the sodium can then be used to power ATP synthase. So they don't need to rely on electron transfer to power ATP synthase in this case. Yeah, very interesting. Next slide. So we have this very complex chemical process we have mentioned about photosynthesis, but now we are going to remove all the parts that we can. So next slide. So we can remove carbon fixation, and also some photosystems to have what we call the cyclic electron transport. So it's photosynthesis is just one form of phototrophy. We can still have a functional system even without carbon fixation. But now we can remove even more components that are absent in methanogens and acetogens. Next slide. So we can remove the quinones and the cytochromes. They are not necessary as well. Next slide. We can, of course, we have in the methanogens and acetogens, we also have no photosystem, of course, but they do have ferredoxin. Ferredoxin is one component of photosynthesis, and ferredoxin is likely the most ancient part, like aside from ATP synthase. I would suspect that ferredoxin is even more ancient than ATP synthase, but that's my guess. But in any case, ATP synthase and ferredoxin are likely the most ancient parts of photosynthesis as well. Next slide. And so now we can further reduce the system even further, and we have something like this. We have simpler homologs of complex 1 and the NAT1 complex found in chloroplast and cyanobacteria. And these use ferredoxin to pump protons or sodium ions, or the process can also go in reverse. They can use ion gradients to reduce ferredoxin as well, and this is basically a very among the most primitive systems of electron transport that is not reliant on sunlight. So again, photosynthesis is not necessary for life to exist. Life can exist without sunlight, using systems like this. Next slide. And the energy gained from this primitive form of electron transport chains, that powers the acetyl-CoA pathway. They can use this to make biomass and then release acetate or methane as the waste product of that. Also, this can go in, as I mentioned on the bottom right there, it can go in both directions. They can oxidize ferredoxins to pump protons and sodium ions, or they can use the ion gradients to reduce ferredoxin, and that ferredoxin can then be used as the electron donor to drive the acetyl-CoA pathway to make biomass. So this is a much simpler system compared to photosynthesis. It is probably the system that existed before photosynthesis evolved. Next slide. So now we move on to the evolution of reaction centers. This is the most conserved part of photosynthesis that we have discussed. If I get around the time to do it, I will discuss the evolution of the chlorophyll pigments and also discussing the evolution of the Calvin cycle. But for now, we will focus on the evolution of the reaction centers. Next slide. So just a recap, this is the core subunits of photosystem 1 and photosystem 2. So photosystem 2 consists of 4 subunits, the CP43, D1, D2, and CP47. And also in photosystem 1, you have you can also say 4 main subunits. You have plastocyanin shown. So, but plastocyanin can also be ignored as of now because it is also not necessary for the function. Like, as I mentioned before, some other bacteria uses cytochrome C in place of plastocyanin as well. Also, interestingly, the unit PSAC is homologous to ferredoxin on top of this PS1 structure as well. So next slide. So now we have removed those structures to make it even simpler. So this is these subunits are the most conserved parts of all phototrophs, including the non-cyanobacterial cyanobacteria as well. Yes. And also these two structures, the PS2 and the PS1 structures, are homologous to each other as well. And next slide. So now we simplify the structure that we have discussed before. Let's see here. I have to remind myself of the notes that I made on the slide. Let's see here. You have the protein structure consists of two domains. You have two domain types. You have the electron transfer core domains, which consist of 5. Let's see here. TMH. I forgot what TMH stands for. Hold on. I need to remind myself.
Well, Nesslig, we are at the 2-hour mark. So.
Do we need to stop?
Yeah, we need to stop for today, and then we can pick this up next Thursday.
Sure. Yeah. I think we we just begun the evolution of the reaction center. So I think we can start a few slides ago and then pick up what we left up next time. Sorry about this.
Okay. No, that sounds great. That's perfect.
Yeah.
Okay. Peter, you want to pull us back out, please?
Yeah. Yeah. So right. I think we will. I think for the next episode we will then go back a few slides and then begin at the point where we will discuss the evolution of reaction centers. And it will probably, like, I'm almost finished. It's just maybe half an hour more.
Okay, sure. Yeah,
Yeah.
we'll do that next time. That's fine. Okay.
Yes.
Thank you, Nesslig. That is really interesting. And so we'll have to finish up with that next Thursday. And so I hope everyone will join us for finishing the evolution of reaction centers. And that's all from me. I haven't really worked on any videos recently. I've been slammed with everything here in the meat space. So, normal job. And then so like I work at the aquarium, and then I teach classes at a community college. And also, fun fact, I'll get to do some lecturing at a museum and in the near future regarding an exhibit that's through the Smithsonian that involves mosasaurs and other prehistoric aquatic animals that have been found in Angola. So I'll get to do some talking about that at a local museum. I'm very excited about that. It'll be at the end of the month. So, okay. Tabula Rasa 0606 says, thanks, Professor Nesslig. And thank you, everybody, for watching, and we will see all of you next Thursday. So, bye, everybody.
Bye-bye.